The box jellyfish larva is obviously a lot simpler than we are. We're not going to learn too much from studying one single system, and we need to move now towards human vision. We share something with the box jellyfish larva, though. We too are products of evolution, and evolution has happened in a terrestrial context on planet Earth. And planet Earth is bathed in the light of a particular star whose light has particular properties. Not all stars are the same. They all emit electromagnetic radiation, but they do so in different ways, with different intensities at different frequencies. What we think of as visible light is just a small part of the electromagnetic spectrum, in which energy is radiated at a diversity of frequencies. And on the slide there, you can see the narrow range of that spectrum, which we associate with visible light. Physically, this is not any special part of that spectrum. The light to which we are sensitive, which we see as light, is an undistinguished part of a continuous spectrum. Different parts of that spectrum we become aware of in different ways. We've developed technologies, for example, to make use of different parts of the spectrum. So TV, radio, X-rays and so on, all use different parts of the electromagnetic spectrum, not the visible light part. The sun emits energy across the entire spectrum, but not all at equal intensities. And if we look at the relative intensity of the sun's emissions at different wavelengths, there's a peak there, shown as the topmost peak in this diagram. Also shown, the bottom peak, is the relative sensitivity of the human eye to energy at different frequencies. And you can see the match, right? Our eyes are not sensitive to random variation. Our eyes evolved in a specific terrestrial environment, and so they're tuned, and our visual system is tuned to our environments. It's inseparable from the environment in some ways. Moving then to the eye, we don't see with our eyes, of course. Seeing is something we do with our entire body and brain, including brain. The eyes are an extremely important part of it. No one's denying that. The eye itself houses the retina, which is where the receptors are concentrated, which will ultimately stimulate neural firing based on patterns of light and dark as they fall there. Now, the patterns of light and dark are further focused and enhanced in some respects by the enclosing body, by the presence of a pupil, which is a narrow hole, and by the lens, which focuses light. Now, it's not helpful to think of the pattern on the retina as an image. You are a product of an image-saturated culture. You grew up with photographs and moving images. You watch screens all day. Images are part of our cultural inheritance. They're not part of our biological inheritance. And human seeing is not substantially different from the seeing of any mammals, and other mammals have no images. Most humans at most times had no images. So don't think of the eye as sending pictures to the brain. It absolutely doesn't, right? It's the start and central point of an entire whole body visual system, and the eye itself is cradled in a dense array of muscles that allow it to orient with respect to the world. So let's have a look at the retina. On the retina we find two major types of receptors, not one but two, and they do very different jobs. There's a small part of the retina on which the incoming light is most keenly focused. It's a small area called the fovea, and when you look straight ahead, if you look at your fingertip, for example, then the line from the fingertip through the pupil would fall on the fovea. That line of orientation is relevant to where you're attending in space. On the fovea, we find cone cells. These are very complicated receptors which are sensitive to color And they're very, very densely packed. There's about 7 million of them on each fovea, but they're restricted to a rather small range. Distributed over most of the retina, we find a different kind of receptor. These are called rods. And in one eye, you have about 150 million rods, so an awful lot more of them. And rods are sensitive to something rather different. They're sensitive to movement, particularly, contrast, but not to color, interestingly. Now remember, you don't see with your eye, you see with your whole body and brain. So it's not necessary to ask, what do the rods see or what do the cones see? Those are not appropriate questions. The retina also has one spot in which there are no receptors. Excuse me. This is called the blind spot. You're not blind there. Rather, this is the point at which the optic nerve emerges and heads on its way to the brain. There's about 1 million nerve fibers for each eye going towards the brain. And it's to that plumbing we're going to turn next. The plumbing is a bit mad. Here's a cross-section through a brain, horizontal cross-section to a brain, and we're viewing from beneath. Those are the eyeballs up at the top of the picture. And you can see the optic nerve, there's a blue bit and a red bit emerging from each eyeball. We'll distinguish the blue and red bits in the middle. Whoever wired this system up made some strange choices, because your eyes are very close to your brain, but they connect to the back of the brain, not the front of the brain. Isn't that weird? That's evolution for you. And furthermore, it's a long way from the eye to the back of the brain. And there's a couple of important way stops first that completely affect and change the character of the stimulation traveling along the optic nerve. First, we have what's called the optic chiasma. And it's at this point that, as you can see in the diagram, some of the fibers cross and some of the fibers don't cross. We'll get to that in the next slide. There are then a few more way stations and branching points before anything ever hits the brain. Again, the back of the brain. So the lateral geniculate nucleus is shown there. There are branches that go off to a part of the brain called the thalamus. And eventually then the first cortical brain connection to the eyes is in the so-called occipital lobe. Now we're going to view this from above. There we've got the two eyeballs looking at a spoon. And if we imagine this head held still and the spoon held still, what you can see here is that the light reflected off the handle of the spoon falls on the left side of each of these eyeballs. Light reflected off the bowl of the spoon falls on the right half of these eyeballs. That's geometry. After the optic chiasma, fibers have switched from one side to the other, some of the fibers and not others, such that those patternings which are happening on the left side of the eyeball now all get processed or progress further on one side of the brain, and all those on the right half of the eyeball progress on the other side of the brain. This means, in effect, that the right half of the visual field is treated first and foremost by the left side of the brain, while the left half of the visual field is treated by the right side of the brain. It's not the case that the right eye projects to the left side of the brain. The distinction refers to the visual field, which is the space in front of you when you hold your head still. The right half of what you're seeing will, first of all, project to the left side of the brain and vice versa. Now, I said the part of the brain that the eyes connect to most directly is the primary visual cortex, which lies in the occipital lobe. So let's have a tiny bit of crude brain anatomy. The brain is a very complicated structure. There are deep structures within the brain, but the part of the brain that you're most familiar with is also, from an evolutionary point of view, the most recent part of the brain, and this is the neocortex. It surrounds the brain on all sides, and it has two halves, two hemispheres, and each half can be distinguished into four main areas as shown on the diagram. They are not actually brightly coloured, unsurprisingly. The occipital lobe is the green area at the back. We've already met the frontal lobe when we were discussing human development. That's the very large part of the front of our brains. The temporal lobe is on either side, just behind the ears, although the plumbing is no easier there than it is for the eyes. And the parietal lobe is the bit on top that joins all these parts. On this diagram, you can also see nested below the neocortex a different structure called the cerebellum, which is like a mini brain in itself. It's old, has an old evolutionary origin, and we'll meet it in a later part of the course. That's enough brain anatomy for now. Now what we're going to do is we're going to meet some work which follows the projections. We've just gone from the eye to the primary visual cortex. Where do you go after that? Well, there's a lot of ways to understand brains, but a very influential view of this comes from Goodale and Milner in a 1992 paper, which identified two separate pathways, one coming up over the top of the head and one coming around the bottom into the side lobes. So the primary visual cortex is area V1, and there we can identify successive stages, and at each stage we can ask, what is it about the seeing that seems to matter to this particular neuron? And what we find is that if you follow the connections that come up over the top in the so-called dorsal stream, aspects of vision that are connected with movement and obstacle avoidance are all seem to be what matters to these neurons. Whereas when we follow the connections that come around beneath and to the sides, the ventral stream, things are rather different. We'll have to unpack both of those. Studying the activity of individual neurons is difficult. We're not quite sure how to read them. But when you look at the variation in the activity of individual neurons in primary visual cortex, you can establish that they are related to low-level properties of the light on the retina. So each local area in the retina projects directly to a local area in primary visual cortex. And in that local area, what matters is whether there's high contrast elements, distinguishable shape, patterns of light and dark, possibly moving, that fall in that particular area of the retina. As we move deeper into the brain, what we find is that the activity of the neuron seems to depend on matters that have more to do with meaning than with patterns of light and dark. From an information processing point of view, this suggests that high-level cognition or knowledge and expectations, and perhaps information from other sensory systems, is being combined as we progress. The dorsal stream and the ventral stream use terms that come from anatomy. Anatomists have the challenge of referring to parts of all kinds of bodies, all kinds of bits of bodies in all kinds of animals. Sometimes if you're dealing with an animal that you're not familiar with, or the brain, for example, it might not be clear what's meant by dorsal and ventral. I've included this slide for your helpful orientation. This shows some of the technical words that anatomists used, illustrated on the particular example of this dog. And there you can see dorsal refers to his back. You may remember the dorsal fin on a shark, for example. And ventral refers to the belly. In the case of the brain doesn't have a belly or a back, but dorsal would refer to over the top and ventral to underneath and around. So, the dorsal stream, following this one. If you don't like the technical term dorsal, you can call this the where stream, because it seems to be associated with where we are in space, what's moving, are we moving, are other things moving? It's very important not to walk into walls. I don't need to remind you of that. And your visual system is useful for not walking into walls. This is the kind of thing the dorsal stream seems to be involved in. That is, when we study the variation in the activity of neurons along this pathway, we find them sensitive to movement. And movement can occur from two sources. It can be your movement in the world or movement of things in the world near you. The ventral stream is rather different. This is the one coming down, and we might call this the what stream because it seems to be involved in identifying things around you. Now, you don't have to identify or name anything in order not to walk into it. You merely have to avoid it. But to name something, you have to recognize it. And we recognize all kinds of things. Faces are one of the most important things that we recognize, and indeed we'll find neurons down here which are sensitive, particularly to faces. The part of the brain that the ventral stream leads down to here is in the temporal lobe. As I said, just inside from the ears. In fact, it's the inferior temporal cortex, shown here in green. And neuroscientists have mapped out the sensitivities of individual cells here, both in monkeys and occasionally in humans. And what they find is that individual units here respond to very complex properties of what there is in the world to be seen. So not patterns of light and dark, but patterns like being a banana or being someone's face. There's no way to map the space of meaning. So neuroscientists going in here and looking at the sensitivity of individual neurons are always tapping about in the dark a little bit. Well, what they always find is that there's organization based on meaning. You never quite know when you examine a single cell here what it's going to be sensitive to. The individual organization is therefore highly individual, but clearly it's complex properties of the world that have been organized here, not low-level properties of light and dark. Let's have a look at one study that was done using a rhesus macaque monkey. They're the workhorse of cognitive neuroscience. In this study, we're looking at two studies here. In the first study, a very, very fine electrode was placed inside one neuron in inferior temporal cortex, recording the activity of that individual neuron. And then the game starts. Neuroscientists have to show the animal various things, get it to look at various things, and somewhere along the line, maybe you find something that the cell responds to. In this case, this cell responded to the face of another monkey, and the top left-hand plot shows you the response. The x-axis goes from 0 to 180 degrees, and that shows the angle of the face of the monkey that the subject monkey is looking at. And you can see that the response is specific to a particular angle. In fact, it only fires, as shown by the histogram and by the blue curve, it only fires fires strongly when the monkey is looking at another individual monkey in profile. That's very, very specific. So a conspecific face in profile is the kind of thing that this is sensitive to. Now, there's no generic pattern of light and dark that corresponds to a conspecific face in profile. This is meaningful response. We can see how we might interpret the activity of such a neuron in the bottom right plot, where after trying many, many things, it was found that a different cell in a different monkey responded to an image of a hand. Great. Once you find something that the damn cell responds to, you can then vary it and see how sensitive it is and what kind of hand. So an image of a hand produces a strong response up in the top left corner of the bottom right-hand figure. You can see the red histogram showing a peak. Turn the hand around, and the peak is still there. So this cell doesn't care whether the hand is this way or this way. It's kind of excited by the presence of a hand. So now the game begins. In the third figure, you see an outline of a hand, just a drawing of a hand, and the cell seems to respond to that. So there's something about hand shapes here, not textures, but shapes. Now, then the experimenters contrast its response to this and its response to this. By changing orientation, you completely change the pattern of light and dark on the retina. The cell doesn't mind, so it's invariant with respect to orientation. It doesn't matter what angle the hand is at, this cell is responding to a hand. If you make the display a little bit smaller, the response goes down. So it does care how far the hands are from the eyeball. Further investigation then shows that it has to be a hand and not anything else. So replace the hand by a mitten and the response goes away. Replace the hand by a bunch of finger-like things that are not in a hand-like form of organization, as in the bottom row, no response. No response to faces, no response to abstract shapes. Now we know a lot about this cell. We know it's sensitive to the presence of a hand, not its texture, but the outline shape of a hand in any orientation, but at a fixed distance from the eye. I have a puzzle for you. Who owns the hand? Pause the video and think about it. Did you get it? The monkey owns the hand. Think of all the hands in the world that you see. Which hands are a fixed distance from your eyeballs? We primates manipulate things in the space in front of us. So there's only two hands in the world that are regularly and reliably to be found at a fixed distance from you, and they're very important. Your two hands. So we have here a cell which is responding selectively to seeing the monkey's own hands. That's very high level and meaningful. Now, as we go up these so-called processing pathways, we find cells that selectively respond to more and more high-level meaningful properties of the world, although nobody has yet found the mythical grandmother cell. This will be a cell that responds only to the presence of your grandmother, for example. But some cells can be very specific to high-order properties of the input, for example, responding to familiar faces. And in the next video, we'll look at an illusion based on a highly specific response to a face stimulus.